Royal College of Arts, Science and Commerce, and particularly microbiology department, as well as our beloved Madam Dr. Surekha Zingle, that she approached me almost like 2 months back. And of course, this Indian Women Scientists Association, as well as BRNS, for selecting me to give a popular talk on the topic that is interest of all of us: cut, copy, paste genes for crop improvement. With your permission, I'm going to add one word. So beyond cut, copy, paste genes for crop improvement. So I will talk a little bit even more on this topic. So I do not want to take individual names. I thank all of the organizers and even the student who introduced me for all your good. Let's see. I think we will come back to business, and we will have more time for interaction later. So what I thought is, today we are going to talk, about this genetic engineering. But initially, I would like to introduce my institute, National Chemical Laboratory, and then I will try to give some idea about natural genetic engineering. But because what genetic engineering we are doing in today's time, it's not something new. And for that, I've been going to talk about 3 major processes that we are doing since our civilization. One is called domestication. I will talk about it a little bit later. Hybridization, that everybody knows that we are breeding the crop varieties and creating better varieties for whatever the desired trait that we want it to have, whether it could be fruit size or color or nutrition or whatever trait that we are looking for. And similarly, we are using also classically this mutagenesis for having a better varieties for whatever the trait that we are looking for. In last 20-25 years, I can say our understanding about functional genomics and genomes has, and maybe a technological development in molecular biology, we can even apply this molecular, genetic engineering approaches to really do the crop improvement. So I don't know what is the issue here. Yeah. Okay. So I'm going to give some examples, since Madam has shared with me some of the syllabus. So I will talk a little bit about these 3 or 4 different genetically modified crops, which are used not in our country but in other countries. For example, rice, brinjal, tomato. And towards end, if time permits, I will talk a little bit about our work in this direction. And this whole of the talk will give you emphasis like why we need genome editing or genetic engineering, particularly for agriculture applications. Okay. So just to give you a brief background of our institute, CSIR-National Chemical Laboratory. So we are part of this Council of Scientific and Industrial Research, where our Prime Minister is the President of CSIR, and Minister of Science and Technology is Vice President. And then we have director journals, governing bodies, and advisory bodies. And there are 38 labs in the country in different areas. And we have almost, 5,000 permanent scientific staff, which includes classical scientists as well as a technical staff who supports most advanced or state-of-art technologies. And in the laboratory, the head of the laboratory is director. And then we have research council as well as management council to monitor our progress and our directions and give time to time different advices. So NCR at a glance, we are established in 1950, almost immediately after independence. At the moment, we have more than 1,500 people working on it, out of which 500 are permanent, around 150 scientists. We have at any given time around 500 PhD students and another 500 research staff who are doing actually research on the paper. The areas that we work in NCL are polymer science, and these are the specialized, branches of chemistry and biochemistry, like polymer science, organic chemistry, catalysis, material chemistry, chemical engineering, and biochemical sciences as well, and process development. So we have excellent infrastructure. I mean, I will say one of the best instruments which are available today in the world are available with us to measure the science and chemical information that present in various systems that we are working on. So major activities where we contribute to the nation is for technology development. We support the industry, we support the, I will say, society, and of course we also publish papers, patent, and on an average we have 50 students getting PhD every year. So the group that I am working is called Plant Molecular Biology Unit, and Dr. Ranjekar, who came back from Canada in 1976, started this group, which is first group in the country who started working on particularly, I will say, plant molecular biology. So during the years, we keep changing our, I would say, areas. So from 1976 to 85, we work on structural organization of repetitive DNA in plants. From 86 to 94, our major emphasis was on plant genome analysis. And then at present, we are working on functional omics. That means we use genomics, proteomics, metabolomics, or bioinformatics, whatever the technologies are available, and to support agricultural biotechnology research. This is very important to say that when this National Chemical Laboratory started way back, this purpose of this laboratory was to actually help the society in the chemical and biochemical sciences for the good of the people. And this was said by our founder, director, Professor McBain. So coming back to the presentation, I thought I will give you first slide. And since all of you are students and particularly coming from the colleges, so if you want to enter into the research, and in my opinion, everybody of us actually have a curiosity, and basically that is, in my opinion, research. We do in more formal way in the laboratories, but I think everybody does the research at a certain point and keep this curiosity going on. So as soon as you think about research, I feel research is beyond borders. Why I say, if you see this picture, Professor Venkatraman, who got Nobel Prize in 2010, if you see his actually a background, he did his BSc, MSc, and PhD in physics. His work was more of to understand the translation process. All of you know, since you are biology or chemistry students, that how the protein synthesis occurs in the bacteria. So he worked on this ribosome and antibiotic interactions and how this tRNA recruits the amino acid. So his research was in biology and Nobel chemistry, or he got Nobel in chemistry. So you can imagine that in research there are no borders. It was not that the Nobel Committee does not know that his background is physics, he worked in biology, and how they are giving him Nobel Prize in chemistry. And this is not an exception. If you say, I just was listing out some of the Nobel Prizes from 2010 to 2020, and all these red you see, these are actually, I will say, areas of chemical biology. That means biology is a strong part in all the red that what we have actually I have put forth. For example, last year's Nobel Prize goes to two women scientists. I'm sure Madam will be very happy to know this, Carpenter and Donda, for genetic scissors, which is called genome editing, or a tool for rewriting the code of life. And we are going to talk about it a little bit more. But all others, like for example, discovery of green fluorescent protein, or structure and function of ribosomes, or G protein coupled receptors, or mechanical studies of DNA repair, or electron microscopy for high resolution of biomolecules, something like a bigger molecules, or even the fast display. So all these areas are very cutting edge technologies where biology and chemistry is work together. And that's what I am proud that my basic training in master's is in biochemistry, so that I do have some background of biology and chemistry, and that's what I joined NCL for my further career. So, so this is a very simplest form of central dogma. I do not want to make it too complicated, but if you see any plant, it has cells, or any organism for that matter, it has cells. And these cells, I'm sure all of you know, there is a DNA, which is a genetic material, and this DNA has genes. Actually, it's like although compared to like a whole genome, only few percent of that has actually the genes, and other is also very important for the regulation. And these genes gets transcribed and translated and gives protein, and these proteins actually give a life to organisms, from bacteria to whatever today's human being. And these proteins also works like enzymes or structural proteins for a number of things, and they also produce metabolites. Particularly if you are thinking about the plant, they produce variety of metabolites, more than, I will say, 10 lakh of different molecules that plant produces. These are called secondary metabolite or specialized metabolites. And not only humans, but all other organisms, it is actually source of nutrients. And also there are some molecules which are toxic to the humans or even the other animals. Also, plant produces very nice, the way we have a defense system, or in animals, the way we have immune system, plant do have a very nice defense mechanism, or also the basic processes, something like cell cycle and regulation. And in my opinion, it is really important to understand these interesting phenomena, because today's time, what we are facing and what this endemic or pandemic, we are realizing that we are really not keeping our nature more safe, environment more safe, and it is really important. And that's why we need to understand the rule of nature. And one of the way I keep saying this to the students, particularly, that nature's law is live and let others live. Unfortunately, we human beings, we feel that only we will live, and that's what we are facing to some extent, some problem. Why I say this in a context? Because when we look to the plant defense mechanisms, I found it is very fascinating, since plants know how to really restrict the pathogens as well as insects feeding on them, but they don't kill them. So they have a rule, as I said, that live and let others live, and try to control their populations and so on. So it is important for you to really understand these definitions. And since your college authority as well as Professor Jingre Madam told me that the students might have this background, but I just wanted to just remind you that please understand the definitions of genes, alleles, genotype, that means genetic content, phenotype means what we see. And then there is something called gene silencing. It's not only the overexpression, but you can even knock down the gene, particular gene, or you can even knock out the particular gene. And also very recently, we called it as a genome editing. Also, there are some more important definitions which you need to really understand at this stage. Is recombinations, which is a process which pieces of genetic material, that is typically DNA, is broken and recombined. And genetic recombination is essentially means the same. Particularly in prokaryotic, there are like this something called horizontal gene transfer. So that's what I try to say, that the genetic engineering is not new. It's not only it happens in higher organisms, it happens even in prokaryotic. So this horizontal gene transfers, we have, substantial evidence that this happens, that organism incorporate genetic material from one to other in a sexual form. Also important to understand whatever that traits or characters that we see is actually contributed by the genes. And these traits are sometimes could be qualitative and sometimes could be quantitative. What exactly it means is like there may be multiple genes involved into giving a particular trait, and sometimes a single gene can also give a particular trait. So these are like, again, the genes are actually controlled by a very typical and very classical way, which called environment. And that's what we call G effect or G interactions. That means genes as well as environment decides what trait has to be there or what trait has to be there, and so on and so forth. And genetic engineering, that is what today's thing is like. It's not only overexpression or it's not only silencing. It means everything, whatever we play with the genome, means the act of modifying genetic makeup of an organism is called genetic engineering. So traditionally, as I said, since we became, I will say, hunters or gatherers, or when we start civilization, since then we are actually selecting the crops. That means we are actually doing the genetic engineering. So this is, we called it as a domestication, or I will say it's called traditional crop modifications. Later on, in last maybe couple of 100 years, we are doing a purposefully mutation breeding, or I will say classical breeding, taking two parents and getting the offspring with a variety of things. And in modern era, we are doing genetic engineering. That means we are doing transgenic breeding and also genome editing. And this is what the picture that I would like to show. All the students, typically, if you want to develop, for example, if you see this is a plant which is having a disease susceptible but good yield, and the other plant, which the cartoon that I'm showing here, which is having less yield but it has disease resistance. So what classically we do is we cross and we get the population, and we select through generations. We call it F1 generation, F2 generation, so to get that character much more stabilized, and you can have actually final plant or final crop variety or elite line that is having both the character, that means having a good yield as well as disease resistant character. And this is typically, actually, I will say, depends upon crop systems and so on, but it takes around 5 to 7 cycles and approximately, I will say, 8 to 10 years to come up with a variety which is having this interesting trait that we were interested in, like having a disease resistance as well as a yield. So this is what we call conventional plant breeding, but it is actually a genetic engineering. As I mentioned to you that when we are looking into it, like we did some specific mutations by using chemistry, our chemicals, or whatever the mutagenic chemicals or radiations, and then we select, and then again we can randomly you have to select through the generations, stabilize that character, and you can produce the elite one. Variety, again, this is genetically engineered, because you, unless you change the genetic, if you come back to the transgenics, so here we might do a precise thing. That means a particular gene take a piece of a DNA which encodes for something, put into the specific modified plants. It takes around 4 to 6 years. If you see, this is a picture. I mean, as I said, that we are doing this from more than 10 to 12,000 years ago. For example, if you see this cartoon again, some plant has a bigger size of crossing, either naturally or purposefully, and you can select and you can get a really, I will say, a plant with having a desired fruit size as well as fruit content. And if you want to show in the cartoon form, for example, this is having a small, I will say, seed size, and this is having a big seed size. You can cross it, but this has more number of seeds, this has less number of seeds. So if you do the classical hybridization, you can mix this trait, you can get a recombinations, or I will say this is called a recombination inbreed lines, and you can select and you can get both characters in a particular plant. And this is what I mean, it's possible, and only limitation here is that these two plants should be crossable, and that's what we called it as a classical breeding. So examples of traditional, actually, I will say, crop modification. So if you can imagine, 7,000 years back, we have such banana having a lot of seeds and a little of flesh, or I will say a very tough part. I'm sure if I give you this banana to eat, I may not be able to eat. But if you see today's modern banana with hybrid Musa acuminata or Musa balanata, you will be able to see very nice fruit. So this is actually happened through the genetic engineering. Although we did not do precisely taking the gene, but we did selection on the basis of something which actually modified the genome, and you get such a nice fruit. It's not only true for one plant, whatever the crop plant that we grow today in agriculture, this is true for all. I'm just giving one or two examples. For example, if you see this eggplant, which is a brinjal, bengan, that's what we say. So it was not originally like this, what we eat today. This is typically like bengan Bartha's time. So we enjoy these, but basically the wild eggplant was having a different colors, was not really a shape that what we are looking for, or it has very tough or hard spines. Whereas in the modern eggplant that we see, which has a desired character and more importantly, the good nutrition. Again, I'm saying this is not true for one plant. Whatever the plant that we are growing today in agriculture, it is not related to India everywhere. For example, the brassica, like cauliflower or cabbage that what we eat, or broccoli what we know, or even other fruits or other vegetables that we know, their original was not like that. So these are, for example, terminal buds, or this is the flower clusters, or these are flowers and stems. So all this, from this to this, or from this to this, happen through genetic modifications. And that's what I repeat, and I keep emphasizing, saying that genetic modifications we are doing more a classical way. Now with the modern era, we can do in more precise way. Mutagenesis, as I mentioned to you, it can be done with chemical mutagen. It can be done with I will say radioactivity or even isotopes and so on. So, but again, this is called induced mutagenesis or site-directed mutagenesis or insertion mutagenesis. And again, it is a modification of the DNA. Okay, so typically what you generally do is you treat the plant variety with mutagen, then you get typically a plant which is actually having some variation. You may not able to visualize this, and this is what we call that as a phenotype. So you may not able to really see in the first generation, but if you continue this generation, the M1 means it's the first generation, M2 means second generation, and so on and so forth. So you'll be able to see these phenotypic characters that you are interested in, probably in the M2 generations. And if you keep selecting this further, you may able to really get the plant that you are interested in. Of course, this is more of a random mutagenesis. You need to have a good eye, and that's why the agriculture scientists in this country or all over the world have some very, I will say, vision to see what characters they are interested in and what characters they can increase the yield and so on. So in genetic engineering, which is in today's time what we call, it's called recombinant DNA technology, is actually we can change the genetic makeup. And a simple example for that is, for example, if you can, so if you know the limitations of classical hybridization is like you can use or you can cross the two plants only in case there is a possibility of crossing. But with modern genetic engineering, you can take a protein or we can take a gene from the bacteria and put into the maize, and you can get, I will say, a bacterial gene expressed in the maize having whatever the characters that it has. Okay, so why this is possible? Because as I said, that we have now a good understanding about the genes. That means what kind of a function particular DNA has. And there is a lot of advancements in the molecular biology. That means we discovered the several enzymes, several vectors or plasmids that we can say. So we can isolate and amplify DNA. We can understand based on the sequence. For example, you know, today NCL is contributing significantly to in our genome of this new variant. So this is what we can really amplify and sequence it. With enzymes, specific enzymes, that was a great, I will say, tool. So that we can cut, then you can paste it. Then there are advantages in transforming, then screening, and then of course you can overexpress. And this is what DNA you want to amplify, again, then you can cut and put into the proper thing, and you can transfer the number. You need to have purification tag so that you can purify the protein that you are interested in. And these are the typically the maps of these vectors, which are very interesting. And again, I don't want to give you information, but you can look into it more details, like how one can do it. So there are very I will say, great discoveries, something called TA cloning. For example, if you see, our tag DNA polymerase has the ability to add A at the end. Okay, and then if you have vector where you have T, which is a base, another base which is complementary to A, and then you can ligate. So this is called TA-based cloning, and this is very well possible through even restriction digestion as well. Another great discovery for the plant transformation is concerned is discovery of this Agrobacterium, which is, we call it as a nature's genetic engineer. It's a soil phytopathogen, which is naturally infects the plant, and this is called tDNA, that means transfer DNA. So this is typically what you see happens in the Agrobacterium, and this Agrobacterium has natural ability to infect the plant and form the galls, and that's how the DNA, whatever the DNA which is present in the form of TI plasmid, can be transformed to the plant cell, and then you can propagate the plant and you can do these steps. And this is again, I will say, cartoon presentation. For example, if you have insert interest of a gene in there, then you convert into or transform into TI plasmid. Then once you have this TI plasmid ready, then you can transform to the plant, and then you select based on your antibiotic resistant genes, and you can get the particular gene which is not there in the earlier in this plant is there. So this is what the natural process. Again, this is a textbook information. I'm sure you will be able to find this information even in the textbook. So nowadays we are talking about more of not only just overexpression or silencing, but you can do a precise genome editing. And that's what last year's Nobel Prize goes to, this discovery of CRISPR-Cas. I will just give you a background of it. Again, this information is available, and there are other methods, something called TALENs, for example, transcription activator like effector nucleases or zinc finger nucleases, or even endonucleases, which are our meganucleases. So that means basically you can particularly get rid of a part of genome or part of the gene, and that's how you can make genome editing possible. And these are like very classical example how we can remove the gene. This is the discovery, last year's Nobel Prize goes to it. And especially this is important to tell to microbiology student, this was a natural mechanism present in bacteria and even archaea to how to really defend themselves against the viruses. And this actually came up with the discovery of this CRISPR-Cas9 and how essentially it happens. I mean, now there is a lot of advancement. You can knock out particular thing, or you can even add something into the genome with the CRISPR-Cas things, and it has a variety of applications. For example, in gene regulation, epigenome engineering, genome imaging, CRISPR-Cas. I mean, there are even, I will say, more than 20 different CRISPR-Cas types are actually available. Recently, we have reviewed them. If you are interested, you can, get this on internet, what different methodologies are available for doing that. So what essentially we achieved with this? For example, all this above thing, whatever you see, whether it is a blight resistance in rice or herbicide resistance crops, or even I will say a disease resistance in wheat, or high yield waxy corn, what we today eat, or soybean, or whatever. I mean, number of things that has happened through this genome editing. High yielding rice, for example, all this is happening in last, I will say, from 2012 to 2020, or even before. People were trying to modify the genome and getting into the particular crop to enhance a particular trait. So what exactly essentially we want to do? For example, modification of a particular genome we would like to do to increase the nutritional content, or have a more tasty food, or its appearance, or even for attractive like flower pigmentation. So that's the basic idea that we do the genetic modification. Why also we do the genetic modification? All of you know that we use chemicals to control the pathogens and insects. So to have a natural resistance in the plant, we can take such an insect resistance gene or insect herbicide genes, and you can introduce in the plant so that you can avoid using these chemicals, so that we don't pollute the nature. So this is also possible. Also, there are like a stress, not only the biotic, but even there is like environmental stress, or maybe if you plant don't have ability to handle the, for example, salty water. So all these traits can be introduced into the plant through genetic engineering. For example, like if you know that some of the fruits have a perishable life, like for example, tomato, sometimes it is 2 rupees kilo and sometimes it is 100 rupees per kilo. So that happens because we cannot store this fruit. So if you can delay the fruit ripening, you can even actually give a good justice to the farmers so that they can get a good value for their. Not only that, there are talk, and since it is included in your syllabus, you can even produce the vaccines in the plant. Of course, there are some limitations for that, and we are going to talk about it, but of course, it will be overcome in a due course. So I just wanted to give a very first example where something from the plant has been actually expressed in the yeast. Yeast is, all of you know, you guys are microbiology student. And from simply sugar, you can produce a drug which is actually used to treat a malaria called caused by this Plasmodium falciparum, and the molecule is called artemisinin. This is not a molecule present in the yeast, but it was present in one of the plant called Artemisia annua. So what people have did, what scientists have did, is they actually arranged this metabolic pathway in such a way that by expressing the plant genes as well as by silencing some of the yeast genes, to have more substrate available for production of this artemisinin. And by expressing these two genes from the plant, one is called this ADS and another is called P450. These two genes can give this artemisinic acid in such a way that this is actually available or used as a drug molecule to treat the malaria, not only in India, but all over the world. So this is what the beauty of, I will say, genetic engineering, if you use a conscious. So this is actually, other example, taking something from the plant and producing in the yeast. And that has also a lot of significance. And today's area, something like chemical biology or synthetic biology can do these things. What we are going to talk today is probably the other way, that how we can improve. And these are the top 10 genetically modified plants. For example, corn, which is having a high yield. Soybean, for example, it has having a good content. The cotton oil, or maybe disease resistance, the resistance against papaya, for example, there are viral diseases on papaya, and this was the first plant, actually food plant, which was released in Hawaii, transgenic plants, where they don't get virus diseases, and rice or rapeseed or potatoes, tomatoes, and whatever even dairy products as well as peas. So all these plants has been actually right now used, not only, not in our country probably, only cotton is approved in our country, but other plants are actually used all over the several parts of the world for various characters which are very interesting, are very safe to use this transgenic plant for. I will give you some example. For example, golden rice. Again, this is other way. The earlier example I gave, something is produced in the plant, but they use the plant genes to produce in microbes. Now this is other way. So they have used genes from the bacteria and other systems to produce this golden rice. And this is the pathway of this golden rice. For example, this alpha carotene, which is actually very important. And all of you know that we have this vitamin A deficiency. So we can have this provitamin produced in the rice by just changing, or by just introducing these two genes, and one can even get a substantial amount of carotenoid, which is a precursor for the vitamin A. Not only that, the advancement in this area can even increase something like even 23-fold of this carotenoid, something like 37 microgram per gram. And this is what the golden rice do, which is actually waiting to use for, I will say, in several countries. And in my opinion, this could really bring, or this could really take care of these nutritional issues. Okay, so same thing for tomato. I mentioned to you, for example, this is the one of the gene which is called polygalactonurase, which is expressed during the tomato ripening. And if you can silence, you can have, or you can increase the shelf life of the this tomato. And by the way, I can tell you that it doesn't really influence the taste as well as nutritional content, but it increase the shelf lives. So these are the best characters one can think about introducing and using even in our country. So BT brinjal, for example, again, for most of the vegetables, we use chemical pesticides to control the insects as well as pathogens. And this BT brinjal was developed in a whole idea that this plant itself will increase the resistance, or it will not get infected by the insect, which is a very common insect on this brinjal. And the BT brinjal was developed. And in our country, it is not allowed to use, but in, for example, in Bangladesh, they are using from 2014, and there is something like 51% yield was increased, whereas the usage of pesticides was actually significantly reduced. And that's something very interesting. I mean, in our country, we are still waiting because we got GE approval, but there are some issues, and we are still waiting that we should get approval to use this BT. So edible vaccines, again, so the methodology remains the same. Basically, you take the gene which gives this particular I will say vaccine or particular protein. You can take the gene of interest, you can infect, and the way procedures are developed for transgenic plants through using either Agrobacterium or to bombardment, you can use, and you can have the vaccines produced in the plant. Of course, this has to go through oral because it is through the plant. So it does work. It may be not possible for all vaccines, but it might work for several vaccines. So there are examples that people have already shown that various vaccines are produced in the various plants, and they are actually can be used for these things. Of course, there are some certain limitations for it. And what are these limitations? For example, the advantage is that it's non-toxic. It has low side effects. Basically, it's less expensive as well as effective. But what also advantage is it can produce in the really, I will say, mass produced. It is economical. It will be coming back to the work that we are doing. And Madam Sureka Madam particularly mentioned me that I should give some examples from the lab. So one of the projects that our 3 projects I'm going to describe very briefly, because I know there are, we started a little bit late and we have a limited time. So one of the projects that we are working is to understand the plant-insect interactions from the native and agriculture systems. So the idea is to use molecules in the form of specialized metabolites or proteins or genes for crop protection. And the insect that we are working is very notorious insects. All of you know that in Maharashtra and few other states, a lot of farmers even commit suicide. This is the insect which is actually responsible for it. It is called Helicoverpa armigera or bollworm, which infects more than 250 different plants and causes very severe damage. So we are working on this, and our idea was to screen the plant which is having a natural resistance against this insect, and we found this Capsicum annuum, chili, or I will say capsicum. It is this insect doesn't feed on it, and we identified some molecules called protease inhibitors. And again, the name itself is indicative of its function. It actually inhibit the digestion of the insect. And these genes that we clone more than 70 genes from this Capsicum annuum, and few of them we have patented. And what was very interesting about these genes is like they will not kill the insect, but they will block the digestion, and that's how they can reduce the fecundity and fertility. That means a typical moth of this larva might lay 1,000 eggs. But if you feed on these protease inhibitors, they lay only 50 eggs. So that way, it's a natural way of controlling the insect populations. You might not get a clean field. And if you see, like this is application of these protease inhibitors on the leaf disc, and you can see that they are protected. And now our idea is, can we develop a transgenic plants expressing these protease inhibitors and try to understand from the nature and learning from the nature and trying to use this more elegantly so that we can obtain the insect resistance. And it is not only that we are doing, this is one of the very interesting paper. For example, they have expressed two protease inhibitors, and you can see typically insect will grow like this in the larval form. But if you use this protease inhibitor, the growth is inhibited. And you can imagine if such growth is affected, it might reflect on their fecundity and fertility, and we can save our crop plants. And this is actually the field trials. For example, this is a plant which is non-transgenic, and this is a plant transgenic with expressing protease inhibitor. And you can see the cotton yield significantly increased. So these are like some of the, advantage of using genetic engineering. Another example I'm going to do you, which has a lot of significance. For example, we work on mango, Alphonso, particularly. So Alphonso has several metabolites. I'm not going to go into detail, but these are the specialized metabolites that we identified, which gives unique flavor to the Alphonso. For example, these lactones and furanones, which are very unique in Alphonso compared to other varieties. For example, we screen more than 27 varieties of that are popular in this country, and found that the chemical composition that present in the pulp that what we eat is different in all these varieties, and particularly Alphonso has this lactones and ketones, which are very, very unique, and they give this specialized aroma. And when we screen this population from 3 different locations of this Alphonso, from the size of like peanut size to fully matured mango, and then different stages of ripening, what we found very interesting is, for example, we collected the Alphonsos from Dapoli, Devgad, and Vengurle, and we found that there is something which is very specialized in the Devgad, that lactone content of the mangoes which are collected from Devgad region are very high. And all of you know that we like to have mangoes or Alphonsos from Ratnagiri and Devgad. So how this happens, why these lactones are again very high in the Devgad region, or very high particularly on Alphonso, this is also called very nice programming. And this is what we have analyzed. For example, Alphonsos, which has having rich in lactones, and that gives a very nice aroma or nice taste. The Pyri, which is another variety which has a low lactones, and I'm sure you will agree with me that Pyri doesn't have a taste like Alphonso, so it has low lactones. And then the variety which is actually a Kent, which is don't have lactones. And if you go back and try to understand how these molecules are produced, and when we go back, there is a nice programming that happens in these fruits, and that's how you get these kind of things. So if you understand this biosynthetic pathway, that how these lactones are produced from these unsaturated fatty acids, you might able to really don't have to do the genetic engineering, but you can lactones in other varieties. So this is the beauty of, I will say, this is what exactly, again, we have several patents on Alphonso mango, particularly these genes which are coding for very, I will say, enzymes that are very unique for a particular pathway, and we are actually working on it. The last project that I'm going to talk to you, this was a collaboration with Israel, Weizmann Institute of Science, Israel. So if just to give you an example, like for example, if you know that these kind of potatoes are not good for us, and when we looked into it, what happens in these potatoes, why this actually starts? So we found there is a light inducible compound called steroidal glycoalkaloid, which is actually when the potatoes get exposed to the light, their concentration goes high, and they could be very harmful for us. There are cases in developed countries like in America or in Australia and Europe that people died by overeating such as sprouted potatoes, and this is a molecule responsible for it. So when we started working on it, both in tomato and potato, we do found that in the ripening tomato, these compounds are disappear because they are present in the green tissue. And we could understood, this was a paper we published in Science, that how we showed that from the cholesterol, how this chromatin in tomato or from cholesterol to alpha-solanin in potato are produced. So there are 10 different genes that we have characterized. And what is the beauty of it? Like once you understand this pathway, you can do the engineering. For example, we don't have to do, I will say, take a gene from other things. So we just silence this particular gene, for example, GAM4, and what we could see that the level of these compounds like alpha-solanin and chacochin are reduced. And then there is some other compound is actually getting produced. And this was very beautiful because this compound, what I showed you in earlier slide, for example, is good for human health. And this paper was published in Science, and then the editor wrote about our paper from nasty to tasty. So if you see more carefully here, the alpha-solanin and chacochin in potato, for example, such potatoes, whether you keep in light or dark, the levels are very high. But if you silence, you don't have to really transfer any gene, just silence particular gene, and you see the levels of these compounds are actually almost negligible. So that's the beauty of, I would say, a more precise building through genetic engineering. So I know I was running with this slide. So this happens with the similar compound, which is chacochin as well, same story. So that means we can use this genetic engineering approach in a very safe way to get rid of even anti-nutritional compounds. So with this, I would like to thank you, all of you, for your kind attention, and particularly the pioneers of this plant molecular biology and our workhorses, like all these students. We called it as a PMB family, plant molecular biology family, started in 1976 by Rajakar, continued by Dr. Vidya Gupta, and right now myself, Dr. Narendra Kadu, and 2-3 young scientists, Dr. Kirti Kumar, Dr. Rakesh, and Dr. Archana. Her picture is not there. So these are the young scientists that we are working together as a family, as a joint family. So, and these are all our students who actually contributed for this project. And I would like to thank all of you for giving me opportunity, particularly Royal College authorities, microbiology department, and in particularly Dr. Surekha Jingre madam. She is always, I will say, inspiring for us. I mean, we know from, each other from last 15 years, and whenever I talk to her, I get a lot of energy and a lot of enthusiasm to continue research. And she's such a great person. It's always a pleasure to talk to her. With this,